All right. Okay, so first of all, a couple of things we're looking at. So we pick up where we left off. So we were looking at Shargoff, and a couple of things I want to touch on just to kind of collect some things together is we want to start off with a bit of the journey of the DNA structure, right? So this is kind of like what sort of grabbed a lot of attention because we felt like we were hot on the trail of the inheritance molecule. So that's really cool. We're getting ready to figure something out. So Avery, McLeod, and McCarty had done their thing. Hershey, Chase had done their thing. And so we kind of felt like we were starting to learn a few things. We also knew that this happens every now and then in science. There was also a very, very clear perception, and I like to say that scientists don't think this way, but they do. We all knew that whoever the first person was to discover the structure of DNA was going to win the Nobel Prize. That was a foregone conclusion. And so, as is the case, you can imagine that there is any number of shameful competition to get there, right? To be the first one across that finish line. And I would like to say that scientists are more noble than that, but we would be lying to you straight out if that's exactly what we said. Scientists are no more noble at times than some of the most noble pirates that we know of. We're going to take a look at a couple of those surly scallywags, so to speak, right? Here in just a second. Just a couple of things. First of all, however, leading up to this journey, this discovery was made, like, it took us about 50 years to get there, but there's a piece of some things to this. And this is kind of like, I like this. There's a lot of tales in these discovery processes of how to do things and how not to do things. And this is another one of those sort of illustrative sorts of things where we had, last time we had kind of like this idea that that Mendel was not in isolation, that he basically built upon his forerunners, in this case, Kohlreuter and T.A. Knight. And I made the point there that science was a communal activity, right? That it's a cultural activity, and that any one person is not endowed with any great potential. They're standing on the shoulders of those who went before them, and oftentimes those who went before them get no credit at all, even though one would argue that they actually made the most important components. I like to think of scientific discoveries like solving a puzzle, a communal puzzle, right? So if you ever had a puzzle like in a break room somewhere and it's just there, and you can sit down in a break and just sort of work a couple of pieces and then sort of do a little bit of stress relief. Or if you don't like puzzles, that would be the opposite of stress relief. That would be stress inducing. Either way, it's up to you. You can sit down and work it or not. And so it's kind of a community project, right? So ultimately, that's kind of what scientific discovery was like. And that's certainly what the discovery of DNA structure was like. So here's a couple of things, first of all, that we want to kind of put together and kind of add that we already know. So the first one is taking a look at the base pairs, right? Because early on, we were able to actually identify these base pairs. We were able to identify them as purines and pyrimidines. We weren't necessarily able to sort of see them structurally and that sort of stuff, right? This came a little bit later, but we did know a couple of things, right? So if you take a look at the actual structure of the bases themselves, notice that you can tease out a little bit of their interactions. In this particular case, we're looking for evidence of hydrogen bonding, right? So what are the requirements for hydrogen bonding? What do you have to have? Oh, this goes way back, right? To the chapter that you guys have probably long since drank away. So water, but what is it about water that forms hydrogen bonds? They go back to the definition of hydrogen bonds. Remember that slide with hydrogen bonds? So that's a different chapter. So that's the next chapter. That's the macromolecules chapter. So polarity, right? So you get into the polarity piece. That's a bit about water, right? So what is actually required for polarity? So you have two different things, right? An electronegative atom and a not so electronegative atom, right? So creating the two different poles, that negative pole versus the positive pole. So generally what you're looking at is something electronegative versus something not, right? Remember the definition of a hydrogen bond. You might want to refresh that one, right? This is an electrostatic interaction between the positive-ish charge of a hydrogen of one molecule and the negative-ish charge of an electronegative atom on another molecule, typically oxygen, nitrogen, or fluorine, but for fluorine doesn't exist in biology. So it's largely oxygen or nitrogen. Why? Because oxygen and nitrogen, if you take a look at them, are electronegative. Hydrogen, not so much, right? So let's take a look at this. So here you can see clearly evidence, not for the negative side of things, but for the positive side, right? Because nitrogen is electronegative, so it's going to make these hydrogens look a little on the positive side. Here's a nitrogen that's electronegative. That's going to look a little on the negative side. And then, of course, here's a hydrogen. It's not necessarily all that positive, but this guy certainly looks like it has potential to be negative, right? Same thing over here, the oxygens. Well, that's a gimme, right? We've already seen this one actually in protein structure. This guy clearly has a negative-ish personality to it, right? Because of electronegativity. Here's an electronegative nitrogen, but it's going to be making that hydrogen look a little positive, right? You're stealing all those electrons from it. And then, of course, here again, you see a nitrogen that's exposing these hydrogens, and then here's a negative there, okay? So all I'm doing is I'm just mapping where these sort of polar kind of charges are on these molecules. So here, again, you have a kind of a positive saying there, negative, negative, and there's a positive there, right? Because of that electronegative nitrogen pulling those electrons away. You see a similar sort of thing here, negative, there's a positive, there's a big negative right there, and there's a big positive right there, right? And then, of course, with uracil, you see a negative there, positive there, negative there, a positive there, very similar to thymine. So now what we have is a handful of positives and negatives, right? So we essentially have what is essentially the foundation. For making hydrogen bonds. Doesn't necessarily mean they're all going to interact in hydrogen bonds, but you certainly have the setup to do so, right? Because you got a bunch of positives and negatives due to their polarity and that electronegativity of those atoms, right? So that's basically the personality of these bases that becomes very important. Why? Because what Chargaff saw was working with these bases. And what Chargaff saw was essentially that the number of adenines always equaled the number of thymines. So if I had 50 adenines, how many thymines would I have? 50, right? He also noticed the same thing about cytosines and guanines, right? So if I had 50 cytosines, I'd have how many guanines? Right, so it's the same, right? So it's one-to-one relationship. That was important because what that was basically saying is that there's a definite, very distinct relationship between these, right? This is a non-random relationship. So the fact that you have equal amounts of each is telling you something. Well, what Chargaff was actually finding is essentially the idea of complementarity. So this is basically, anyway. So what this basically means is the reason why you have the same number of A's and T's is because every time you have an adenine, it forms actually two hydrogen bonds with a thymine. It's its partner. And whenever you have a cytosine, it forms three hydrogen bonds with a guanine. So the reason why you have equal numbers is because they form in pairs, AT pairs and CG pairs, and then we later called these base pairs. So when you were doing electrophoresis and you were looking at those bands on the gel, those were base pairs, certain number of base pairs, right? So the small ones had fewer base pairs, the big bands, the ones that ran slow, had more base pairs, right? So because you were working with DNA, you were looking at the numbers of AT pairings and CG pairings. Okay, so those are the number of base pairs. This was really important, right? Because it's starting to get you that sense of relationship, the sense of how basically the DNA is held together, right? The fact that these two bases actually kind of zipper up to each other with these hydrogen bonds. That was an important concept of interaction. The other thing that we also want to sort of lay the groundwork for is the idea of DNA being a helix. Now, there's another character in this chase who kind of had a little bit of a head start, a brilliant guy by the name of Linus Pauling. Now, Linus Pauling was basically the discoverer of alpha helix from secondary structure fame from proteins. And so that's his thing. So he was the one who discovered alpha helix and the structure of alpha helix. As a matter of fact, that's what he won his first Nobel Prize for. He actually won two. He got the second one for peace because he became an ardent anti-nuclear proliferation proponent in an era in the 50s, which was very dangerous to do so. You can get yourself thrown in jail for being a suspected communist. Yes, we did actually do that. Yeah. Just ask any of the communists who survived from that era. But anyway, or you got deported if you were a non-citizen. So he was already hot on the trail of a helix. So structurally, he would be considered like the expert, right? Like if you wanted to learn about helical structure and what the structure of a helix looks like, it's Linus Pauling. He's the one you would go to. So he had a huge head start, as it turns out, on this race and chase for DNA structure. Why? Because as it turned out, in the lab of Maurice Wilkins was a woman by the name of Rosalind Franklin. What she did was X-ray diffraction studies on DNA in this particular case. And essentially what X-ray diffraction is, it's kind of like taking a molecule like of DNA. It's got to be ultra pure, right? So it's just DNA, that's it, nothing polluting it. And then you basically beam X-rays at it, and then you would analyze the scatter pattern that you would collect on the other side. It's not unlike sort of me having a light behind you shooting at me and me trying to figure out your structure by your silhouette. Okay. Now, obviously, your structure is going to have a different diffraction or a bending of light, in this case, X-rays. And so that'll be indicative of your structure. So the pattern you cast is a one-to-one relationship to your structure. So as it turned out, Rosalind, in her X-ray crystallography studies, was able to determine that DNA was helical, although it wasn't the kind of helix that alpha helix was. It was a little bit different. It was weird. They didn't understand it exactly. But she was able to determine that DNA had a diameter of about 2 nanometers and that it completed a turn, a helical turn every 3.4 nanometers. So she was able to provide a lot of critical sort of what I call statistics of the actual helix itself. The problem is it wasn't a single helix like what alpha helix is like. So it's a little bit more complex, which is the reason why it kind of stumped us a little bit. And this is the actual crystallogram in question. Now, what that means then is the second that you pin DNA as a helix, what that does is that immediately puts Linus Pauling at the head of the pack. Because after all, he is Mr. Helix. And so he was very, very close to winning his unprecedented third Nobel Prize, which he probably would have won had he actually crossed the finish line. But the problem is nobody told Linus Pauling that he was competing with cheaters, which is exactly what they were. Because as the story goes, Rosalind is nowhere mentioned in the discovery of DNA structure, at least in the literature of the time. There's only 2 names that you hear about. These 2 guys. That a little passive aggressive? I didn't actually intend it to be passive because these bastards, as far as I'm concerned, can be expunged from the history of science. There, is that a little clearer? Yeah, it actually gets worse than that. I've been known to drop F-bombs in this particular section. But here's how it goes. And the story is depending on there's different versions of the story. But basically as it goes, Rosalind had basically this X-ray crystallogram of DNA. And the most aggressive anti-Watson and Crick story suggests and strongly implements, with absolutely no punches pulled, that basically Watson and Crick moseyed down the hallway, got a hold of Rosalind's X-ray crystallogram, and pretty much used that information to solve the structure of DNA, and then go immediately to the presses that they had discovered DNA. Essentially, they stole her data. There's no punches pulled. That's not an implied. That is an explicit accusation. Is that they outright stole her data, which in scientific culture is akin to putting a noose around your neck and basically kicking your own chair out from underneath you. You will never be considered a decent human being again. Oh, but it's worse. The softer versions of this will kind of soften that a bit and say, well, they didn't actually steal her data. They were kind of collaborating. They were talking about the data and they saw the data. And then that kind of solved that last little bit of the puzzle for them. And then they were able to come up with the structure of DNA. So it was actually more of an honest collaboration. They didn't actually steal it. Whatever. Personally, I prefer the former rather than the latter. And here's the reason why. Because even if it was a passive ripoff or a collaboration, what happened afterwards is actually as galling as potentially what might have happened beforehand. Because after the fact, this was so sensational that all the press and all the media attention all went to these guys. These guys in the scientific world achieved a level of stardom that was unknown to scientists at the time. Scientists were largely obscure, nerdy individuals rattling around in the lab. Most people didn't know them. And these guys were front page news everywhere. They were rock stars. Culturally, societally, they were out there. They were celebrities. They wrote books chronicling the discovery of DNA molecule, of course, making themselves look like the messianic prophets that they are, right? But guess what? Do you hear anything about Rosalind? No, as a matter of fact, actually, you get what is essentially a systematic burial of Rosalind. So not only was Rosalind not mentioned, she was not credited, she was basically aggressively annihilated from the conversation. In a classic, and this was a visceral thing that just happened here, right? In a classic male chauvinist type of domineering sort of a way. And it was brutal and absolutely unacceptable. Matter of fact, this was for science, and I'm not trying to hyperbolize or cheapen it, but this was kind of a Rosa Parks moment in science and scientific culture, where it was like the touchstone that just kind of like after this, generations of scientists coming up were absolutely livid. I mean, pissed off, angry. We still are. You know why? Every single textbook has Rosa Parks, has Rosalind R's, right? So, and actually the first four letters are the same. But anyway, right, has Rosalind's picture in it. Every single textbook. Why? Because succeeding generations were absolutely beside themselves with anger over what happened to Rosalind Franklin. Because she was buried, and it was pretty obvious why she was buried. She was buried because she was a woman. That's why. Matter of fact, Watson, of Watson and Crick fame, was a notorious racist. He was actually fired from multiple institutions. So that dirtbag, I'm glad, is gone. Crick, who probably should have known better and could well have been aiding and abetting a racist, which is still guilt, by the way, just letting you know that. He actually went on to contribute. To his credit, he did add a few things, but still, there's a reason why both of them get the goatee, right? Because there's a couple of things you never do in science. Number one, You never steal somebody else's data, ever. Right? I mean, we will kill you if you do that. It is personal and visceral. This is the reason why generations, we're not just talking about a few people. We're talking about generations across the US, certainly, and across the international scope globally. I mean, for generations, we are now probably about 3 or 4 generations of scientists removed now from this. And we are still, to this day, you can see it. Most biology instructors, it gets personal and heated when they talk about Rosalind Franklin. You know why? Because Rosalind Franklin is us. She is us. And Watson and Crick are basically the white privileged bastards who think they own us and they can bury us. That's why Rosalind Franklin is personal, because every single grad student coming up can identify with that. There's these guys at Boulder who've won Nobel Prizes, Tom Check. But if he takes your data, guess what? He just Rosalind Franklin'd you. So that's number one, you never steal somebody else's data. Number two is you never, ever not credit somebody else for their discovery. Notice what happens now. When somebody wins a Nobel Prize, almost always they say, listen, I got the prize, but I just want you to know that if it wasn't for this lab over here and these guys over here and these guys over here, none of this would have happened. They almost always, because they almost always give credit to everybody else in the industry. Because that discovery that one person got the Nobel Prize for, that one person almost never did it. You know, there's a third thing that's also galling in science. We've already got 2. Any one of those 2 is a career killer by anybody else's standards today. The other thing that is actually more galling is basically you got the rainbow and the pot of gold for everybody else's sweat equity. Everybody else worked their arse off. And you just sort of sat there and sort of gleaned the rewards for it. You did nothing, you lazy pieces of crap. In science, when you win that Nobel Prize, the reason why we have less problem with it now is when we take a look at them, we know them well, first of all. But almost everybody who wins a Nobel Prize, you look at them and you look at their career and you're like, yeah, you put in your dues. You didn't just show up yesterday, do some sort of an experiment, and here you are today. No, you've been at this for decades. You've put in the dues, right? You've put in the work. You've been there sweating right next to us. Yeah, does it suck that I didn't get it and you did? Yeah, of course it does. But at least I can take solace in the fact that, you know what, man, you've been there. You were right there next to me, digging that grave, if you will, so to speak. In the case of the Neanderthal genome, literally, you were digging the grave, right? You're digging them up, but right, I mean, you were right there next to me. And so I saw the sweat and the blood and the tears coming out of you, just like it was coming out of me. So I'm okay with that. These guys did nothing. These guys were wastes of skin. The equivalent, let's go back to the jigsaw puzzle. The community's putting together the puzzle. Everybody puts together pieces. And then guess what? Here comes some schmuck right at the end, picks up that one last piece and says, hey, look, there it is. I just solved the puzzle. Look at me. Aren't I great? I'm going to write a book about how I solved the whole puzzle. And I'm going to go on tour. I'm going to have speaking engagements. And I'm going to have just about any university appointment that I want. And I'm going to be a rock star. But guess what? Do I talk about the female who put most of that puzzle together? No. No. It's character assassination. That's basically what Watson and Crick did. That's the reason why it's galling. There's 3 cardinal sins that you never commit in science that Watson and Crick committed all 3 of them. And so they are the example of what not to do. Because I can tell you right now, Watson and Crick would have done nothing if it weren't for Rosalind. They would have done nothing if it wasn't for Shargoff. They would have done nothing if it weren't for Linus Pauling. Because actually, they were just empty heads. That's all they were. And the fact, and whenever I see all their notoriety in their books, it just makes me want to vomit. If there's ever there is a reason for a book burning, right there, right? That's one of them. Okay. So now we know how that goes down. You notice I skip over the slide. I have to, because the second I catch that one right there, that one, I'm ready to punch somebody. Because basically you walked off with the premier prize for doing absolutely nothing. Pieces of crap. Their entire names should be expunged from history. Why? Because a single individual never makes a discovery. Science as a culture, as a group, makes the discovery. And we each solve the the puzzle together, one piece at a time. Now, how many seats are on the Nobel Prize? There's 3. It can be shared by 3 people. Think about it. What should have been just? What would have been the right thing to do? Okay, fine, Watson and Crick, whatever. I questioned that one. Anyway, they shouldn't have been there. But okay, whatever. Watson and Crick, if that's what you want to do. And who should the 3rd seat be? Absolutely Rosalind Franklin. But guess who got it? If you're thinking somebody with a Y chromosome, you'd be correct. Maurice Wilkins. Nobody talks about Maurice. He wasn't a bad guy. He was just the lab that Rosalind worked in. And Rosalind was like a postdoc. So she was like a researcher. And I know they prattle on about law, but that's not typical culture. The PI gets the credit for all the data. It's their data. And Rosalind, this is just normal. So, no, because you know what? If somebody here, I know that why that's a no. There's a guy in genetics, which we all know and love, right? Because we're all going to take it. But anyway, named Sturdivant. We don't actually cover Sturdivant in genetics because he's a little higher level genetics. But Sturdivant basically, as an undergraduate, he was like a junior or something like that, took the data from his PI, Calvin Bridges, another big guru of genetics back in the 20s. And he basically single-handedly figured out how to map genes. Sturdivant did as an undergraduate. Sturdivant is a male, a white male undergraduate, and his name is in the textbooks. He got credit. So don't give me this bullshit about, oh, well, Rosalind didn't get credit because it was Maurice's lab. Sturdivant got credit. The difference is he had a Y chromosome instead of an X. So don't give me that bull. Anyway, that's that. We're still angry, as well you can see. We're still very angry. By the way, we are doing better as an industry. You see women getting the Nobel Prize much more regularly now. But it was a long time in coming. Because at the time Rosalind Franklin was getting slighted and buried, there was also other brilliant women, one of which was in genetics that I know of, probably the smartest lady in genetics, the smartest person, not lady. I'm not going to qualify it as gender. Probably one of the smartest people in genetics, Barbara McClintock. And the reason why I love Barbara is because she's a corn person. I'm not a corn person. I'm just going to say I'm not a corn geneticist. That's not my thing. I'm more of a fly guy. But my PI at UC Davis, Scott Hawley, who's now in the Academy of Sciences, he knew her. And he mentioned actually Barbara was like so smart that she could actually walk through a field of corn and she could actually tell you genetically what was happening in each one of those things of corn. She was like beyond brilliant. She was like so smart that she was outthinking any of her male colleagues at the time. Yeah, literally. I mean, she discovered some things in genetics that were just jaw dropping. A lot of times things that's like we didn't really believe until a guy discovered them. And then we're all like, oh, look, isn't that interesting? And then, wait. I do believe Barbara figured this one out like in the 40s. Right? I mean, this is a lot of this started coming in. As a matter of fact, Barbara was one of the first women since Marie Curie. The male chauvinists always point to that one. But what about Marie Curie? She got a Nobel Prize. She wouldn't have gotten a Nobel Prize if it hadn't been for Pierre, whom we all think of sort of as her partner. In reality, he was the water boy. Okay, so Marie was the brains of the operation. Pierre was just sort of the dude who just held stuff for her, right? I mean, that's how that worked out. But you had to have a guy in there in order to give the girl, and that's sick. But that's the way, unfortunately, it was, and that was wrong. Barbara McClintock, and I believe she was like 83, they finally gave her the Nobel Prize. Finally. And they would have probably given Rosalind the Nobel Prize, but she died. And so they don't award it posthumously. So that is just, and that's still, it's just a chafing. You can see it's still very much that grain of sand in the oyster. And especially as the generations went on, because I mean, we would work in labs. I mean, just like coming up, being trained. I mean, and we would work for women PIs, and it wouldn't be like a big deal. It'd be like normal. And I mean, these women were brilliant. And it's like, we knew in the industry, it's like, oh my gosh, it's like the women in this industry are brilliant. Why aren't they being recognized like the men? And so that's continually what fuels that. And that's still there. There's still a lot of fire in there. So it hasn't really gone away, as you can see. So not too bad. Only some mild swearing. One of the reasons why my YouTubes are also always selected as not for kids, especially like not for my kids. Because guys, listen, I mean, yeah, I'd rather you not hear your dad getting a little out of control. Huh? Yeah. But yeah, I would have to Disney it up a tad. It's like, okay, it kind of loses its edge if you got to Disney it, right? Because that's what Disney does, right? It takes important things and just pretty much turns them into a joke and a caricature. That's pretty much. Yeah. So here's basically our double helix, right? So if you take a look at this, a couple things I want to pull out of this. First of all, we've already seen our phosphodiester bond. I don't want to beat that one, but oftentimes you want to make sure you see the backbone, right, has a repetitive sequence. So you have your phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar backbone. And then, of course, you have your nitrogen bases sticking out. So if you, sorry, it's dog hairs coming off of me. So basically, if I draw a backbone, so this is my sugar phosphate backbone. I'm going to put a few nitrogenous bases out here, like so. And let's go ahead and put some things in there. So A, C, G, T, A, G, C. How about that? This will be our 5' phosphate over here, our 3' OH over here. So let's go ahead and make the complement to this. So what would go with A? T, and we would have 2 hydrogen bonds connecting it, right? What goes with C? That'd be G, and we'd have 3 hydrogen bonds connecting it. What goes with G? And then we have 3 hydrogen bonds connecting it. And what goes with T? And we have 2 hydrogen bonds connecting it. And then A, T, right? So we got 2 hydrogen bonds there. And G pairs with 3 hydrogen bonds. And then C pairs with G with 3 hydrogen bonds. What's the complement to 5'? What? 3, yeah, right, 3'. So that'd be a 3' OH over there. your 5' OH over here. Now notice, it's anti-parallel. So your 5' to 3' direction is going this way on this strand. It's going this way on the opposite strand. You have a north and a southbound lane, right? So that's what's called anti-parallel. So your DNA molecule is anti-parallel in nature. That's important to understand, right? Because basically, if you're only moving in one direction, like 5' to 3', which is the case in this particular situation, we'll talk about that a little bit later, then that basically means you can only move in that direction on one strand or the other, right? So you can't go back. So here's your DNA molecule. So here's Rosalind's diameter right there, and here's her turn helix at 3.4. And then you can see Chargaff basically added the relationships between the base pairs in there. And so Rosalind then also added the helix component of it as well. So notice what we have here. We've just described the majority of the molecule, haven't we? So what really did Watson and Crick do? It was already there, right? Linus Pauling couldn't finish it off. Actually, I rather wish he had, to be quite honest with you, because that would have been awesome. Because Linus Pauling was crazy as a loon when he was older, but he was a brilliant guy. He was truly a brilliant guy. He would have been one of the few people that I would say deserves a third Nobel Prize. Don't know if they would have given it to him, but he would have deserved it. I doubt if we're ever going to see that again. There's several 2 Nobel Prize winners. Most of them are winners for their discipline and usually for peace. That's usually what it is. I've never seen anybody win it twice for their discipline. I don't know that the Nobel Prize Committee would ever give somebody a third Nobel Prize. They're not the way they are. That's just, they're Swedes. And so they just like, equity, and they're the neutralists. So they want to make sure they share the wealth, which I agree with, honestly. No, a single person shouldn't win everything, right? Because a single person never has a lock on everything. So this is basically just kind of like what we see with the actual hydrogen bonds. So you can see how this oxygen here, that negative-ish oxygen, is going to see that positive-ish hydrogen that's going to form a hydrogen bond. This positive hydrogen is going to see this negative nitrogen, so that's going to form a hydrogen bond. This positive hydrogen is going to see this negative oxygen, so that's going to form a hydrogen bond. So you can see your 3 between cytosine and guanine. And then for the adenine, thymine, you see this positive-ish hydrogen against this negative-ish oxygen. So that's going to form a hydrogen bond. Of course, you have this negative-ish nitrogen because of electronegativity against this positive-ish hydrogen. That's going to form a second hydrogen bond. So you can see the 2 hydrogen bonds there between A and T. Now, a lot of times students will ask questions about this pair down here, right? So wait a minute, isn't there a negative-ish on that oxygen? Well, yeah, there is. And isn't there a positive on this hydrogen? Well, no, not really, because that's carbon, not nitrogen. And carbon, even though it's close to nitrogen, doesn't have as much electronegativity, although technically speaking, it could. Right? So even if this was, and you have a negative over here on this nitrogen, it's too far away in order for those two to interact. So what you would have to do literally is you'd have to rock this molecule down like this, but then you'd break the top 2. And as it turns out, the top 2 are actually a stronger pairing than the bottom. So that's the reason why it's kind of up the upper level instead of the bottom level. Okay, so DNA replication. Let's go ahead and take a look now at the actual mechanism of DNA replication. Now, in the 50s, we realized that DNA replication can occur with 3 basic models. The first one is what's called the conservative model. And this is basically where you're going to make a new DNA molecule from scratch. Right, that's the conservative one. So they're basically saying that when the parental, or this is the old molecule, remains intact, they're saying that what you're doing is you're making a new molecule from scratch, from nothing. Well, there's some problems here. The question is this. Where does the information come from for the new molecule? Remember, this is replication. not creation, right? You're not making something from scratch. And that's exactly what the conservative model is asking you to do. It's basically saying if you're making a DNA molecule from scratch, you're essentially trying to recapitulate what happened back in, molecular evolution, which by any standard took a very long time to put all these nucleotides together randomly. And you're asking that to happen again. And not only that, but you're asking that to happen again. How many times do you go through replication? A lot, right? So you're asking the same process that took a long time to come together the first time to happen multiple times over. Is that actually going to be legitimately happening at all? No. So basically this, when you think it through, obviously this one is almost like just logically off the table, right? However, we leave it on there because we actually haven't disproved it, right? So it is a hypothesis. However bad of a hypothesis it is, we still have to disprove it. The other one is the semi-conservative model. This is basically where you have one old strand acts as a template for the new strand. So the daughter strands have like half old, half new. Okay. And then the dispersive model, which basically is kind of like mixtures of new and old, like a quilt, right? Almost like a Harlequin's costume, right? It's just kind of patchwork kind of a situation, like confetti, basically, DNA confetti. This was problematic because I'm not quite sure how they explain how that happens mechanistically. But it's a hypothesis that really needs to be vetted. And you need to do your due diligence, right, in this particular case. Clearly from this trio, when you think it through, there's only really one that makes any sense at all, right? First of all, I mean, you don't have a mechanism to explain the confetti approach, right? That's dispersive. You don't have the reality to explain or to expect the first one, right? I mean, because if it happened once, it's never going to happen again because the probability is so preposterous that you're not going to catch lightning in a bottle, right? And this is honestly how a lot of times evolutionists will sort of explain the ridiculous probabilities of getting all these things to happen in sequence. They always start off and say, well, but we are the endpoint of that probability. It's like, well, wait a minute, you have billions of dollars of cash, so therefore you must have won the lottery. How did you get the lottery number? I don't know, because I have the money, so therefore it must have happened. Well, that's fine, but that means you're never going to do that. You're never going to do that twice, right? And this is what this is asking for. It's asking you to win the lottery twice, and that's never going to happen. Okay. Huh?
Unless you rig it.
Yes, yeah, unless you rig it, right? And that's a different discussion, right? So, yes. So this is conservative. So here you basically have the old, and the old stays intact, and you basically build the new from scratch. Okay. Now that's got some predictions that we can take a look at, and we'll take a look at them here in just a second. Compare that then to the semi-conservative, where your old will basically separate and act as a template for the new. So here you have a combination of old and new, right? Here's old and new. So old, remember, is the pre-existing one. The new one is the new one. And then of course you have your dispersive, which is just this mix, this quilt, right? So this is kind of like, huh? I mean, this is the one that I always like, wow, this was the loser out of the gate, right? I mean, this is the horse that stumbles out of the gate during the Kentucky Derby, and then they have to put him down, like before he even stumbles 10 feet ahead of him. Okay, maybe they don't have to put him down, but maybe just stumbles, right? He lives. I'm just like, if you're gonna put him down, just give him to me. I mean, I'll take a horse. I mean, I'm not a horse person, but I mean, I'll at least take him. At least he's alive. Even if all he does is just run around a corral somewhere and eat sugar cubes. I mean, I'm okay with that, right? Right? I know. I'm just saying. And knowing my wife, he'd be like a fat horse in like no time at all. She loves to feed her animals, of all types. But anyway, so here's what Messelson-Stahl did, and this is another big experiment that happened because this was the one that sort of teased apart which one of these basic mechanisms of replication was at play here. So just to kind of give you an idea, because I never miss an opportunity to plug history whenever I see a date. Okay. 1958, what's going on there? Well, first of all, if you're in the US, you were still deep in the middle of the Red Scare, right? So if you were anything close to communism, probably even openly liberal, you were labeled as a communist. You probably spent some time in jail. I mean, they were still hunting people down at this time unfortunately in America. Who's in power at this point? Eisenhower. This is the beginning of Eisenhower's last 2 years, right? He's about ready to have midterms at this particular point for the end of it. And of course, Nixon was his vice president. The important thing about this one is this is kind of like the Leave It to Beaver era, right? This is still America in its sort of repressed, sort of white privilege, white picket fence kind of era that the 50s get a lot of heat for. But this is also, at this particular point, there is a young man from Massachusetts who is currently a senator by the name of John F. Kennedy at this particular point, who is probably gearing up his presidential run, which he did 2 years later. And of course, who did he run against? The vice president, Nixon. Right? Eisenhower said, I've got enough, I'm done.
I don't think that's.
Which was smart, right? Because Eisenhower had a good run and he did what he needed to do. He basically kind of rebuilt post-war America into sort of something more functional. So that was what we needed him to do, and he did it. But 2 years later, Kennedy runs against Nixon, and it was, a lot of people think Kennedy slammed it, and he did not. It was a very close election, but Kennedy, of course, the more charismatic one, won and beat Nixon. Of course, Nixon, if you're following history, became president 10 years after this, in '68, and largely fled in disgrace, right? The whole Watergate thing. But the reason why I mention this is because we are on the cusp of, in America, what we oftentimes consider a before and after event, right? If you take a look back at generations in history, the Kennedy administration does clearly mark a kind of a sort of a before and after event. There was America before Kennedy, which is associated with repression and Red Scare and Leave It to Beaver and white privilege and all those sorts of things that we are all now sort of trying to deal with and sort through. And it goes further than the 50s, by the way. I'm just saying, it's just was very obvious in the 50s. And then you had an after, right? There was an after Kennedy. And so there was very much kind of a before and after effect. And Kennedy was a pretty big deal. That's basically what's happening here. Okay, that's the world that we're living in right now. In comes Messelson and Stahl. And so what they wanted to do is they wanted to tease apart the difference between conservative, semi-conservative, and dispersive. And so what they did was they used bacteria, which you tended to use back in those days because bacteria was easy to grow, it's easy to deal with, nobody cared about it. You can kill it off in the droves, nobody cares, right? And you can learn a lot. And so what they did is they said, okay, wait a minute. We want to differentiate between old and new. That was a big problem with this, right? Was like, well, wait a minute, if we're making DNA and we want to know how it's made, then we fundamentally have to be able to differentiate between old DNA and new DNA. If we can't differentiate between those two, we have no hope of being able to figure this out. So what did they do? Well, what they did was something actually quite clever. They utilized N15. So N15 is basically heavy nitrogen. So it has an extra neutron in it. You notice nitrogen is normally at 14. So there's an extra neutron in it. And so what they did basically is they incubated the bacteria in heavy nitrogen. So bacteria would go through their whole process, going through replication multiple times over, just let them grow, let them be happy. But what they're doing is they're building all their nitrogenous bases with nitrogen in them with heavy nitrogen. And so all the nitrogen in their system is built off of heavy nitrogen. And so then what they did was then after they felt like the bacteria were nice and heavy, then they switched them to normal nitrogen, which is actually light nitrogen. And then they took a look at what they got over several replication. Now, the turnover time for bacteria is about 19 minutes. It takes about 19 minutes to go through a cell cycle. So in about 19 minutes, you can get your first replication. In about 40 minutes, you can get your second replication. And so that's what they wanted to do. They wanted to take a look. Now, what I want to do is I want to back it up here, and I want to sort of sketch out what our expectations are before we move forward to Messelson Stahl's results. So here's basically our predictions, what we expect to happen. Now, if your old heavy, right, is heavy because it got all nitrogen 15 in it, and your new is light, then after one replication for the conservative, your first replication would be broken into either heavy or light. So after your first replication, you would have this one, light, and you would have this one, heavy. So the first one would look something like this. You'd see two bands of two different sizes, one light, one heavy. Now, what happens if you sent it through another round? Well, the other round would basically be light, 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 and that last heavy that's still stuck there, right? So then what you would see is you would see a darkening, more light coming on, but you would see a diminishing heavy. That's what you expect to see in the second round. Now, what about semi-conservative? Well, in semi-conservative, you're going to see a third band. Medium, right? Because basically you're going to have heavy light hybrid and a heavy light hybrid in that first generation, which is going to give you an intermediate. It's not heavy, it's not light, because it's got a mixture of the two. So in the first one, you're going to see an intermediate band. Now, what about the next round? Well, you're one of these, like if you use this guy as a template, the light, you're going to have light, light, right? If you use this one, you're going to have heavy light. Same thing over here, light, light, heavy light. So now all of a sudden, you've got an intermediate, but now you have light. And as you keep going, light will slowly over time replace everything, and then you'll see light getting heavier, getting darker and darker and darker. So that'll be the only band, right, that's left. But you expect to see one band in the first, two bands in the second, right? Because you see the light coming on. Now the dispersive, whatever, you expect to see two bands all together. Why? Because first of all, you've got the heavy left over, but here you've got an intermediate, right? So you got an intermediate first band in the first, and then eventually what's going to happen is you're going to start to see that same intermediate over time, right? So it's going to be a slightly different pattern because of the dispersive effect. And part of the problem, like I told the other section, is one of the problems with this is when it's dispersed like this, when it's a patchwork, like the question always comes up, well, how much of this is there? Is there a little bit or is there a lot? Well, if each molecule has a slightly different variation of that, some a lot, some a little bit, then you would imagine you have all these different variations in size. So you're not going to see a clean band. You're probably likely going to see more of like a smear because you have a whole bunch of different variable sizes in there. Okay, that's probably what you're going to see. So what did they see then? So they basically took out their results. And then what they saw at time 0 was one band. Then after one replication, they saw one band, and then they saw two bands. So what did they see then? So they saw something that looked very similar to semi-conservative. First of all, let's take a look. Conservative model basically says that you should see heavy and light, right? But that's not what you see. So gone, you're out. The dispersive says you should see one band in the first and one band in the second, but that's not what you see, is it? Gone. For what you actually see is one band in the first and 2 bands in the second. Guess what? That's predicted by the semi-conservative model. So here you can see this first one is the heavy. So this is the heavy one by itself. After one round, you've got the mixture, right? The one heavy strand, one new strand. So it's the intermediate, right? So this is the intermediate one. And then after the second round, you've got the intermediate, but then you have that one light one. So this is an intermediate plus a light. So Messelson-Stahl's experimental results were consistent with semi-conservative. Now we can put the others to rest. So what does that mean? So what semi-conservative basically means is that we know that the 2 original parent strands are separated and they're utilized like a template. That solves a couple of problems, doesn't it? It tells us where the information is coming from for us to make our new strand. And that makes a lot of sense, right? Because if you take a look at it, the 3 things that you need to make a copy of anything is the same thing if I make a copy of quizzes for you guys. What do I need? I need the actual original, right? So that's one of them. Then what else do I need? So the tool, like the copier machine, right? So the thing that actually does the copying, and the materials that the machine needs in order to copy it, paper, toner, things like that. Those are the same 3 things you need for replication. You need something to copy. Your original. That's your DNA. That's your parent DNA, your DNA molecule. Then you need something to do the copying. This is your copy machine. In this case, it's the replication enzymes that are doing the job. And then you need the materials in order to build your actual copy. In this case, it's the nucleotides. So the nucleotides would be equivalent to like the toner and the paper. Okay. You have those 3 together and you have everything you need in order to do replication. So let's take a look at our mechanism for replication. Actually, this isn't just for replication. This is true actually for replication. It's also true for transcription, which is going to be the next chapter, and translation, which is also next chapter. So all 3 of these mechanisms, and actually many mechanisms like this in molecular biology, have a similar 3-step mechanism that all its activities fit under. So initially it has an initiation phase. This is basically your preparation. So all those events to prepare you to actually do your copying, right? So there's like initiation events. And oftentimes, depending on what you're looking at, whether you're looking at transcription or translation or whatever, then what you're really talking about are the details of the initiation or the preparation tasks that you need to complete in order to get yourself set up to do this particular job, right? And so you can imagine that the initiation events of DNA replication is different from transcription, which is different from translation. But they each have their set of tasks that they need to do in order to get themselves set up to do this. That's initiation. Then once you prepare for it and everything's in place, now you're ready to actually do the job. You're ready to hit start on the copy machine, right? So for me, my initiation events are grab my original, right? I have to stick it in the paper feeder. Then I have to go to the screen. I have to select the number of copies. I have to select whether or not I want it to be 1 to 2 side or 2 side to 2 side. I have to pick which paper, which bin to select from, if it's tray 3 or tray 4, whatever, depending on my paper size. And then I have to select whether or not I want it stapled or not. Those are all initiation events. I'm preparing for the event itself. Okay. Then you get to elongation. You hit start and then away it goes, right? So the machinery does its thing. And so this is basically the enzymes doing their job, the actual task, in this case of replication. And then of course, every good thing has to come to an end. And that's where you get to termination, right? So there are sequences and mechanisms in a lot of cases, some are more clear than others, where you have a certain strategy to actually stop, okay? And so you don't want to just continue on. And so just like with anything, there is a termination. So remember, this 3-step system is true for all mechanisms that we look at. So there is a transcription, initiation, elongation, and termination. There's a translation, initiation, elongation, and termination. And so the events that oftentimes we talk about are essentially events that fit within those three. Okay. So if you want to make these processes a lot easier, always think of it in terms of initiation, elongation, and termination. And that helps you to really kind of keep things straight. Okay. So this is kind of what's happening. This is what we're asking basically in terms of elongation. So here you can see you've got your template strand over here. And then of course you've got your new strand that you're building. And then you can see you've got your 5' phosphate over here and your 3' OH. This is your growing end, right? So remember, this is your growing end. So this is the hydroxyl group that's involved in the dehydration synthesis. And we have the components of water in order to be able to do the dehydration synthesis, which is the reason why that 3' end is the one you grow off of, because you don't have the components of water on the 5' end, right? The phosphate is not capable of making that water product that you need from the dehydration synthesis. So in this case, what's going to happen is your enzyme is going to come over here, read your template. Oh, I see you're an adenine. I know that an adenine always pairs up with a thymine. So what does it do? It grabs a thymine. And then what it's going to do is cleave off these terminal 2 phosphates. That's going to leave as what's called pyrophosphate. And then it's going to make that link. It's going to exit the water. So that OH is going to leave as a component of water. And then what's left behind is the bond that will then link together that last nucleotide. So this is the one you just made right there. So now you're ready to read the next one and do the exact same thing. So you can see, as long as you have template to read, and you have a growing end to reach off of, then you can continue to just add nucleotides to that growing end. Yeah. Correct. So the nucleotides typically are kind of all over the cell. There's usually a reservoir in the nucleus itself. However, like for instance, if you like stick the lysosome on an old organelle, like a mitochondria, for instance, or something like that, with some DNA in there, and you want those nucleotides, or you needed them from your metabolism, where you turn some of those metabolic intermediates into nucleotides, then you can transport those through the nuclear pores. So you can always shuttle exogenous or extranuclear nucleotides into the nucleus itself. But for instance, when you're going through replication, and this is probably a little bit of an unsung idea of G1, because we always think of G2 as sort of the preparation phase. But one of the things that you'll see is that in G1, when you make that commitment to the cell cycle during that G1S checkpoint, then one of the things that you'll get ready for is to get ready for replication. So if that means you need to stock up on nucleotides, you bring some more in, then you start doing that in that particular phase, and then you kind of roll over into the S phase to start making the DNA. If you run out, which sometimes happens, then what's going to happen is you're going to stall out and you're going to abort mission. So, and you can restart though, because you'll still have the growing end there. You can still jump back on there as long as you replenish those nucleotide sources. So that's actually, like, if you were in like a molecular genetics class or something like that, and you were really studying this very closely, we would have a chapter where we would study like the metabolic cycles of nucleotides, so we could really clearly see, like, okay, how are we manufacturing nucleotides? We're consuming a lot of them, that's certainly true, so we're absorbing a lot of them, but like, where are our reservoirs? We're not going through replication. What are we doing with our extra nucleotides? So all of those different metabolic cycles you would study more in depth, like if you're in a molecular genetics course, because then you kind of add that chapter to the story. Now, who's doing all this work? Well, one specific protein, right? So it's the first of our pantheon of enzymes who's in charge of replication, and that's DNA polymerase 3. So DNA polymerase 3 is typically responsible for making our DNA. But let's talk about a few more, because one of the things we understand is our DNA polymerase, which is kind of the one who's doing most of the work. But there's some serious limitations to it, right? So for instance, DNA polymerase 3 can only synthesize in the 5' to 3' direction. Why? Because of the 3' OH. Because that's the only way that you can extend off, right? So there's only a 3' OH over here. So you can only go that direction. right? You can't go this direction. I mean, you can, but that's typically not the direction you're going, because the idea is this one's already intact. That's the template. So that one's already intact. So you're not making a copy of that one, right? You're making a copy. This is your copy up here, right? That's the one you're making. Okay. So that's a limitation, right? That basically means you can only go in the forward direction. Remember, if you're antipolar, that means you have an issue with the other direction, right? So you have to figure that one out. But because you need this 3' OH, this growing end, what that means is you need a way to get a hold of that growing end. So first of all, DNA polymerase cannot self-start. What do I mean by that? It can't provide its own 3' OH. So here's the problem. If you have this piece of DNA, here it is. DNA polymerase is looking at it and be like, great, I've got template. Great. I can't do anything, right? Because I need a 3' OH to start it going. And I can't lay that one down myself, right? So I'm stuck, right? So what you need then is you need a bit of a starter. And essentially that starter requires you lay down a primer of RNA. Now, what is a primer? This is basically just a short sequence that's complementary to your DNA template. So for instance, this guy right here is an RNA primer. So you can see in this case, it's very short, right? So you have 1, 2, 3, 4, 5 different base pairs. That's it. You don't need a lot. You just need enough to provide an OH group right there. As soon as you provide that OH, guess what? Polymerase ready to roll. Can I identify that? And then you can fill in the rest of that. And you're good. But you need that first 3' OH to get it started. And so this primer, which typically is an RNA polymerase, is the one that will set down that initial stretch to create that 3' OH. Now, the important thing about RNA polymerases that are different than DNA polymerase, we'll learn more about this actually in the next chapter, we talk about transcription, is these can self-start. These can lay down a nucleotide just from scratch. Okay, so RNA polymerases are different than DNA polymerases. Now, then what you end up getting is a molecule that's mostly DNA, but this little piece right here is still RNA. So you got to deal with that because our DNA is not a patchwork of DNA and RNA. It's all DNA, right? So that means you have to deal with that one. Is that a question you're saying? Yes, and they'll still be able to pair up with adenines. Yeah, just like a T would.
And then you said that since there's still going to be RNA left, DNA polymerase.
Well, no, actually, they take out everything because remember, the RNA sugars are different. So everything about that RNA nucleotide is different. So the entire thing has to go. But the idea is you kind of have it. It's kind of like a stopgap measure right now. I mean, it's covered, but you got to go back in and sort of do some finishing pieces to it. And we'll talk a little bit about that here in just a second. Okay, so lessons from bacteria, right? So this is basically something that we always do, right? We're always starting off with our lessons in prokaryotes because they are the simplest version of, so if we can't learn it in the simplest system, then the question is, where can we learn it? And the answer is, well, probably not, because it gets more complicated in eukaryotes, right? So learn it here first, and then transfer that knowledge to eukaryotes. That's what we did. So let's take a look about it. There's a lot of overlap here, right? A lot of review here because we talked a little bit about this in chapter 10, right? When we talked about binary fission and bacteria. Remember that the E. coli, which is our first initial model, has a circular genome to it. And we also learned in chapter 10 that E. coli has a place to start replication, right? So it's got that origin of replication, and it's also got a termination point, right? So we learned about that one. So basically you begin your replication at the origin of replication, and then you move in both directions, clockwise and counterclockwise, until you finish. And then ultimately you have two. So this is kind of what it looks like. So here's your origin. Right on the other side, you've got your termination. And so your replisomes will basically bind to the origin, and it'll start rolling in both directions, counterclockwise and clockwise, until they hit the termination point where they hit the connected magician's ring stage. And then at that point, they separate into two separate genomes. And of course, here you see you've got the cell that you go through cytokinesis at this point. Sound familiar? It's binary fission, right? We basically covered that in chapter 10. But now a couple of things. First of all, we're focusing now on the replisome. Now the replisome, you see this term, it's a generic term. Usually you'll see this kind of some term like nucleosome or replisome or spliceosome or things like that, transcriptome or something like that, some sort of an ohm sort of a sound on it. Usually what that refers to is a big complex with lots of proteins in it, but we're not really focused on all those individual proteins. We're just focused on its overall job, which in this case is replication, right? So we know there's a lot of different proteins in it, but we're not focused on that yet. So when we're just talking about the general complex and its general function, we use a term like this, replisome, something like that, okay? But that's usually referring to a big complex. So there's obviously a lot of proteins in here. And so what we're gonna do is we're gonna take a look at what some of those proteins are doing by first stopping and talking a little bit about some of the polymerases. There's more than just one polymerase, which you would probably guess because we started off with polymerase 3, right? So the big question is, well, what about 1 and 2? Well, this is about what about 1 and 2, right? So DNA pol 1 is a polymerase whose job it is to remove those primers. So getting back to your question about removing the primers, this is what DNA pol 1 does, and then it replaces it with DNA. So that kind of gets to your question about how do you go about replacing those primers? Well, pol 1 just basically takes the entire kit and caboodle out, and then there's a gap there, and then it just basically fills in the gap, okay? So that's what pol 1 does. Pol 2 in bacteria, remember this is in E. coli, pol 2 is a DNA repair. So basically what this will do is it'll replace damaged DNA. So it's strictly a DNA repair polymerase. So it's kind of a specialist, if you will. But the heavy lifter is pol 3. This is your main enzyme. This makes the bulk DNA. So that's making the majority of it. Now, a couple of things about these. Their activity. First of all, we already know one activity they have. They have the dehydration synthesis activity, right? They can basically add nucleotides. We already know that. They all have that one. So this one is more of its exonuclease activity. Remember, its nuclease activity is basically its ability to do hydrolysis, right? So these are hydrolytic activities. So when you take a look at all of these guys, all three of them have a reverse gear. They have a 3' to 5' exonuclease activity. What does that mean? That means if you've got 5' to 3', remember it's building in this direction, but what this means is it basically got a reverse Pac-Man. It has the ability to chomp from the 3' end going backwards. So this is like your proofreading, or I like to think of your backspacing. So how many of you guys, when you are typing an email, you flub up the word the, which I do all the time. For some reason, those 3 letters, I just cannot get those right. Right? You flub up the word the, and what do? Hit the backspace button, right? Erasing it, and then you just lay it all down, right? Because it's a fairly easy one to do. So I'd rather not choke up spell check or sound like a complete idiot when you're sending a text. Which, by the way, texts make us all sound as dumb as dirt, even on the best day. It's like, along with autocorrect and all that sort of stuff, it's like, okay, wow, I am not brilliant here. But anyway, right, so basically this is your backspace button. So a lot of times the polymerase will make a flub, catch it, and then it'll backspace with this exonuclease activity and then lay it back down again. Wait, is that a question? Hydrolytic activities. Yep. Because it's already got dehydration activities, right? It builds stuff. So this is its hydrolytic activities. Now, only one of them can actually go forward and backwards. So it's kind of like, and that's DNA pol 1. So in pol 1, basically what you have is you have a dual directional Pac-Man. So you can chomp in the 5' to 3' direction and in the 3' to 5' direction. So this is kind of like your backspace. And this one's like your overstrike. Which I know we don't really use all that much anymore, but back in the day, that's your overstrike button. Go ahead. So, well, that you're talking about the polarity. So we're talking, so in this particular one here, I'm just talking about the one you just made, right? Yeah, so like if you have your 5' to 3', so you're going in this direction, and it's less of a strand as much as a directional arrow because your DNA is going in this direction, and so you're laying it down. And so then you have the ability to do a 5', 3', but backwards, like a backspace, basically. So you can kind of chop that up going back, and then, but only a short distance. So like you're just like with backspacing, like if you basically screwed up an entire paragraph, you're not going to backspace that one, are you? Because that'll take like what, 20 minutes to do that. So that's going to be a different thing, right? Then you're going to be moving to a different phase of cleanup, right? Like a DNA repair, like for instance, a excision repair. So you're going to basically excise that one, cut it out because it's damaged, and you're going to replace it with something a little bit more intelligible, right? And so that one has both forward and reverse. So it can go forward and backward in terms of chomping. We'll talk about that in just the next step. That's as we sort of introduce you to the cast and crew, so to speak. We have an enzyme whose job that is. Right. Well, that's what these guys are. So restriction endonucleases are related to these exonucleases. The only difference is an endonuclease cuts in the middle of the DNA, whereas an exonuclease cuts on the ends. It does, but if you don't have that restriction site in here, that sequence specific site in here, it doesn't have anywhere to cut. Okay. We've already been introduced to a couple, right? Pol 3, Pol 1. Now we have 3 more that we're going to get introduced to. So the first one is helicase. This one is what I call the unwindase. And the separase, not to be confused with the anaphase promoting complex separase, right? This is the separator, right? So you have to unwind your DNA, which is what you're talking about. That's what helicase does. It'll take your double helix, it'll unwind it, and it'll actually separate it. And it separates it into single strands, creating what we refer to as a replication fork. And there's your 2 templates. So the helicase, that's its job. So it goes through, unwinds, and separates. Now, that's one of the major initiation events, right? You haven't done any replication at this particular point. All you've done is unwound the DNA. So here's the problem, though. Single-stranded nucleic acid has one particular fate in the cell, and that fate is gets degraded. So if you have a single-stranded nucleic acid in your cell, its destiny is to get degraded. So all those exonucleases and endonucleases will go after it and chomp it up. They're very active, they're very aggressive. Why? Well, here's the reason why. Your DNA is double-stranded. Not a problem. Safe. Most DNA that especially comes from viruses is single-stranded. So when your cells see single-stranded DNA or RNA, they think, viral genome, let's attack and destroy it. The idea is if you attack and destroy the viral genome, then it will never have a chance to hijack your cell, and you'll never get infected. So what do you have to do then? Obviously, you need to have single-stranded DNA, right? You need to get access to it. So how do you manage to avoid getting digested by your own exonucleases and endonucleases? Well, you have a lot of different strategies to protect your single-stranded DNA. RNA is a master at that, actually. But in this particular case with DNA, we use single-stranded binding proteins. So what these guys will do is they will stabilize and protect the single-stranded DNA. So what does stabilize mean? Well, first of all, as soon as you open this up, they don't want to stay open. Immediately, what they want to do is they just want to Velcro back together, right? So basically, if you had nothing other than a helicase, it would unwind it, and then guess what happened? It would just zipper right back up before you even had a chance to lay down any primer. So you kind of have to hold it apart so that you're going to get a chance to work on it. The proteins doing that job are the single-stranded binding proteins. They maintain the open nature of the replication fork so that it doesn't just re-anneal and twist back up as if you never existed, right? Right. So the helicase would be like, oh man, I just opened that up. Now I got to go back there and open it back up again, and then this one closes, and so it's an effort and futility. Helicase will just be going back and forth, opening it up, and as it goes and opens up something else, this one closes, and it's got to go back, and then so it's going to be going crazy, right? So the single-stranded binding proteins are in place to keep that open and also to protect your single-stranded nucleic acid. The third member of the cast is topoisomerase. So basically topoisomerase is a protein, and there's actually a series of proteins like this, like DNA gyrase, for instance, which will come in there and prevent supercoiling, which basically is this right here. And the best way to understand supercoiling in DNA is to imagine you have a rubber band between your fingers and you're rubbing back and forth. What's going to happen to that rubber band? It's going to get all toiled up. It's going to start folding on itself, right? You get real. What's causing that? Well, the friction of your fingers, true, right? But then what you're doing is because you're twisting a closed system, it's creating a lot of torsional strain. And that torsional strain has to sort of relieve itself, and it does so by creating all these crazy loops and flops and things like that. That's supercoiling. So what a topoisomerase does basically is it comes ahead of the helicase and it nicks the DNA ahead of the helicase to relieve torsional strain. So for instance, your supercoiling is over here. So your topoisomerase will be here, and it'll just basically nick the DNA. Not a lot, just enough, and it'll relieve that stress, that torsional strain. Just like if you take your crumpled up rubber band, all I have to do to relieve the strain, take a pair of scissors, give it a nick, and what happens? All the stress is gone, right? All that torsional strain is gone. That's exactly the same thing as happening. So basically the effect of topoisomerase is as the helicase is doing all this unwinding, it's creating torsional strain in the molecule, which is creating these sort of looping things that are happening, just like a rubber band. And then the topoisomerase is going down ahead of it, and then it's kind of nicking it a little bit, relieving that strain, so it kind of smooths everything out so that the helicase can kind of come through and smooth it out. And you're probably thinking, well, wait a minute, is that a problem? I mean, why would you want to nick your DNA? Is that a bad idea? Well, not really, because why? Because your polymerase is getting ready to come through there anyway. So you can just fix your backbone nick at that point. It's a super easy thing, right? There's actually a separate protein, another cast member that we'll be introduced to, who actually, that's its specialty, is nick sealing. Okay. So now we've met DNA pol 3, we've met DNA pol 1, we've met helicase, single-stranded binding protein, we've met topoisomerase. So we have 5 members of the crew. So these are the players that are essentially going to be part of the replication mechanism, which we discovered was a semi-discontinuous mechanism. Okay, so what does that mean? Well, let's explain what continuous is before we talk about discontinuous, right? So continuous basically means that once you lay down a primer, your polymerase can grab onto that OH and just go and never stop. It'll have free open template in front of it for as long as it wants to go. And so it continuously lays down new template, right? New strand until you basically drop off the chromosome. If you're a linear chromosome like us, or if you come to the termination site like you do in prokaryotes. Okay. So that's what we refer to as the leading strand. It is a continuously producing strand. As soon as you get that started with the initial primer, it just goes and goes and goes and goes. The lagging strand is a little bit different. It's discontinuous. So what happens is you're going to need multiple priming events, and you're going to be basically making this in chunks. That we refer to as Okazaki fragments. So basically, if you were to take dividing DNA and then kind of heat it so it falls apart, everything falls apart like 96 degrees. We saw that with the PCR thing, right? So that kind of basically melts it, right? So denatures it. And if you were to run that on a gel, you would see the heavy template fragments, right, up at the top. But then what you would see is you would see all these little fragments, these chunks that are called Okazaki fragments that are associated with this discontinuous side of DNA production. So what we saw actually was not fully continuous, not fully discontinuous, but a little bit of both. One strand was continuous, the other one was not. We call the continuous strand the leading strand. We call the discontinuous strand the lagging strand. So let's take a look at what it looks like. So here's our DNA. We're going to open up the replication port. So here we're going to lay down our initial primer, right? So there's our 3' OH. So our polymerase is going to grab onto that and it's just going to go and go and go and go. All right, so our helicase is right about here, unwinding everything and separating it. So this is your continuous side, right? Because you can just basically go and go and go and go because the fork is opening up in front of you and you've always got a source of new template in front of you. So you're just going, you're just trucking along. As long as you've got nucleotides and as long as you've got everything you need, you just go straight, right? This is your continuous side of things or your leading strand. The other side, however, is discontinuous. That's the lagging strand. So here, for instance, you're going to make a primer just like you did in the other one. You're going to grab onto this 3' OH just like you did in the other one. And you're going to go in the 5' to 3' direction just like you did the other one. But the problem is the new primer or the new sequence that's opening up is opening up behind you, not in front of you. So you're kind of like moving through all of your stuff and you finish and you're looking back and you're like, but the new stuff is back there. So I have to go back there now and make the new DNA. But here's the problem. I don't have a reverse gear, do I? I can only go forward. So what do I do? So what I have to do is basically I have to fall off and I have to go back to where that new beginning part was and then fill all this in. So when you take a look at it, this is called the lagging strand. This is what happens with lagging strand. So basically what we have here is in the continuous model, this is your leading strand. It's like, I'm going to draw a line. And when I draw this line, you can't just look at the line. You have to actually follow what I do. Okay, so you have to pay attention to what I'm doing. I'm going to connect these two dots. I'm going to do it in a continuous fashion. I'm going to put my pen right there on the dot. So there I am. And I'm going to move forward. Continuous, leading until I stop. So that would be like drawing the line in a continuous or leading strand fashion. However, what about discontinuous? This would be the lagging strand mechanism. I'm going to take the same two dots. Only now in this case, what I'm going to do is I'm going to put my pen right here. And I'm going to move forward because that's the only direction I can move. Then what's going to happen is I'm going to pull my pen off. And I'm going to put it back down right here. And I'm going to connect to the front. And then I'm going to pull my pen off. I'm going to connect right here. And then I move forward. And then I'm going to finally pull my pen off. And I'm going to connect here. And then I'm going to move forward. At the end, can you tell the difference between the two lines? No. But when you watched it, you can tell I drew them very differently. Right? One in a discontinuous fashion, the other one in a continuous fashion. And that's essentially the way the leading strand, the lagging strand actually works. Okay. So we already talked about helicase, right? So basically you open up and you create the replication fork. Now what you have to do once you have it open and you have it stabilized, your single stranded binding proteins, right? So that's already there. Now what do? Well, the next step is to prime it, right? And we actually have an RNA polymerase whose job it is to lay down a primer. And that is called DNA primase. There's another cast member. So we've met DNA pol 3, pol 1, we've met helicase, single-stranded binding proteins, topoisomerase, and now we've met primase. Primase lays down, because it's an RNA polymerase, it can do this from scratch. It lays down an RNA primer that will provide the 3' OH for your polymerase. But wait, DNA polymerase, right? Because now we have an RNA polymerase in the mix. We're going to make sure we differentiate which one we're talking about. So your DNA polymerase needs that 3' OH, and that is supplied by our RNA polymerase, which is our DNA primase. Okay. So it gets a little confusing nomenclature-wise. Just remember, DNA primase is the name we give to an RNA polymerase that's specifically designed to prime for DNA replication. Okay. Okay, so let's take a look then at the actual polymerase itself, right? We're gonna look at leading strand synthesis. So we have one priming event, and then all of a sudden pol 3 just takes over. Now, one of the big things we had in the 50s and 60s was a really strong rush to discover the structure of pol 3. And a lot of the good thing is we discovered a lot about pol 3. We discovered that it was basically made up of a bunch of subunits, right? And that there were several different subunits involved. The beta subunits formed what we refer to as a sliding clamp, which is kind of like shown here. Basically, a sliding clamp is just kind of like you just sort of clamp it on top of your DNA and it just kind of slides back and forth. Okay, so that's the reason. So the sliding clamp model is very accurate. So it's basically sliding along the DNA like that once it clamps onto it. Now, we'll see that sliding clamp piece a little bit later on. The mechanism gets a little busy, but it's actually quite instructive once you kind of get all the pieces straight. So before we get to that big mechanism, we want to break down. The leading strand is fairly straightforward. Yes, I think you prime it once, you put the DNA pol 3 on there, and then off it goes, right? So not only that, but in the leading strand, remember, this is already primed, right? There's your primed event. So there's your 3' OH. So what happens is your sliding clamp will kind of grab onto the DNA and sort of escort the DNA pol 3, and then basically it'll start moving in that 5' to 3' direction until it stops, right? So you only clamp that down, and you basically allow the polymerase to sort of slide. It kind of, in a lot of ways, kind of reminds me, when I think about it, of like the little slide bar on the little weighted scales that you get on, and you kind of move the weight back and forth, you're sliding it back and forth. That's kind of what it's like. So then what about the lagging strand, right? Because obviously this is the trick. This is the tricky one. This is the one that takes a little bit of kind of head wrap a little bit in terms of trying to figure out what in the world's going on with this thing. So first of all, we already know it's discontinuous. We already know that pol 3 has to be involved because it is the one that's laying down most of the DNA as well. We know that we have primers involved, which are made by primase, and they prime essentially what's going to become the Okazaki fragments. We also know from studies on Pol 1 that Pol 1 will come in and remove the primers and replace it with DNA. The other thing we also know in the last one, this is another player that we have, is that those backbones nicks are sealed by ligase. This is what it does. It's the reason why when your topoisomerase goes down the line, nicking the DNA backbone, it's not a big deal because you got ligase in there that'll go in there and just seal those nicks once you're done. Okay. So ultimately then, this is your cast of players. In this case, DNA ligase was kind of the last of the major players. So we have pretty much everybody we need in order to wrap our heads around lagging strands. So here it is. So we already got our leading strand, so we're going to put that one aside. But now you have basically open template, right? So this is an old primer up here that's already kind of done its thing. This is primase laying down a new primer. So this is a new 3' OH here. And so you can see various states here. So DNA Pol 1. So DNA Pol 3 basically is already just finished laying down this segment right here. It's fallen off. And where's it going? It's going to go find that next primer upstream that's behind it. Okay. Now it's already filled it in, but notice it doesn't fill in right there. There's a little gap there. That's the reason why these fragments come off as little small fragments, because they're not connected. Right. So the Pol 3 doesn't connect it right there. Why? Because Pol 1 will come in here and you can see he's basically busy removing the primer and replacing it with DNA. So it's extending off of where Pol 3 left off and then starting to chop up and remove that RNA primer and lay down DNA. And then eventually what's going to happen is your DNA ligase will come in here and seal those individual little nicks. Notice the polymerases don't seal the nick backbones. They just lay down that last nucleotide in line. There's going to be a gap there in the backbone because they can't seal the gap. They can only add to that. So they continue to add to it. What would happen is you would have the strand, the backbone, that would start to go off, and it would kind of like be this little trailing piece next to it. So it would look something like this. So if it kept going, it'd look something like that, right? That's not functional. So what it does is it stops right there and it leaves that little gap right there. That's the reason why you have ligase, which comes in there and seals that nick and makes everything good. While that's happening, your Pol 3 is now landing on this site. It's found a new 3' OH, and it's going to fill this in until it hits the other primer in front of it, and it'll drop off. And then by that time, guess what? You've probably opened up more fork here, and so there'll be another primer for it to go back to. Okay. So that's basically your lagging strand synthesis at that particular point. So when you take a look at your replosome, you can see you've got a lot of different pieces to it. So when we're talking about the replosome, we're talking literally about this cast of players, right? Helicase, single-stranded binding proteins, topoisomerase. So all of these guys together, working together as a complex. This is all part of your replosome. So you can see that it's basically going to be broken into two main pieces, what's called the primosome, which is looking at events of initiation, right? So for instance, primase, that's laying down your primer, helicase, the unwindase, the accessory proteins like single-stranded binding protein, that's what's in this group, and topoisomerase. So those are the accessory proteins, right? And then of course, then you have your complex of DNA Pol 3. Go ahead. No, it'll wind up. Yeah. So once it kind of zippers back up, then and you let it go, you're not stabilizing it, then it'll wind up. And a lot of that is because of the structure of the pyrimides and the purines. Because if you notice that the base pairs are always a purine A with a pyrimidine T, right? Or G purine with a pyrimidine C. But because they are double ring structure versus single ring structure, structurally, they're a little bit different. So it's kind of, they got a little bit of a hitch in their giddy up. It's not a perfect, right? So they kind of got like this little bit of a kink. And that structural lopsidedness naturally causes them to sort of orient themselves into this sort of helical spiral. Okay. So this basically is the mess. So let me explain a couple of players here that are going on, and what we'll do is, because it's going to take a few minutes to do, so I'm going to take a couple of minutes just to sort of point out who all the players are, because they're all here, right? All the entire cast is here right now. So I want to just point out where they are, introduce a couple of people we haven't been introduced to just yet, lay the groundwork, and then what we're going to do is grind through the mechanism at the beginning of Wednesday. So here you can see gyrates. That's in that topoisomerase family, right? Going ahead of everything and smoothing everything out. Here you can see the helicase busy at work, unwinding the DNA and separating it out. And then you're binding to the single-stranded template, these single-stranded binding proteins. Notice they stay there until they are replaced with some sort of a template, right? For instance, primase, which is the next major step, is removing, so those single-stranded binding proteins get removed and replaced with the primer, which gives you your 3' OH, right? And so you can see then the pol 3 will then basically replace those single-stranded binding proteins as it kind of goes through. So here you can see a couple of major players. Here's your sliding clamp. I'm going to just talk about the leading strand. So this is your sliding clamp, your beta subunit, which is clamped onto your DNA pol 3. Now, this guy here, the squid in the middle, is what's called the clamp loader. So it's got basically an area where you have the beta subunit, which actually gets loaded by the clamp. So the squiddly portion is actually what's called the clamp loader. So it'll basically grab onto the clamp and load the clamp onto the DNA. That's what it does for a living. But notice the clamp loader is also attached to everything else in the system. It's attached to helicase in a couple of places. So it's guiding the activities of helicase. It's also attached to primase. So it's guiding the activities of primase. It's also attached to the DNA pol 3. So it's guiding the passage of pol 3. And as a matter of fact, it's also loading the clamp when it needs to get reloaded. So the squid, the clamp loader, is basically the great conductor of all of it, with a hand or a tentacle on every single member of the team, guiding it in its activities, right? So the clamp loader is a really important piece. You can also see the activities of pol 1 down here, replacing primer, and of course DNA ligase down here as well. Okay. So what I want to do is, that's the whole cast. That's kind of what's going on. Then what I want to do is I want to take a look at the mechanistic underpinnings as we sort of move forward, and how the whole lagging strand, the drop off and reassemble works in the context of the great conductor, the squid-like clamp loader in the middle. Okay. So I'm going to start off with this particular slide as a review, and then we're going to move into the mechanisms of that. And we should probably finish chapter 14 pretty easily on Wednesday and get a pretty healthy start on 15. The clamp loader is part of the replisome. So it's not technically the polymerase.
My head is melting. So much writing.
So much writing.
We got to the
Sorry.